All right. Good afternoon, everyone. Thank you for taking the time to join this part one of our FluidForm summer webinar series. And before we get started, I want to remind everyone that for the next 3 weeks, every Thursday, from 1 to 2 p.m. Eastern Time, we'll be hosting the next parts of our seminar series. So with each of the webinar focusing on different aspects of bioprinting. So for next week's seminar, we'll be talking about how we worked with one of our partners, Lulzbot, which many of you might know, to develop a bioprinter that is uniquely suited for fresh bioprinting. We'll walk through a demo and then also go from beginning to end, from designing to getting a print out of the support bath that we print into, and talk about what you can do with the print. For part three, we will really focus in on bioprinting vasculature. What's the mindset when we think about designing a print for it? What are some of the applications for it? What are some of the challenges associated with creating this vasculature print? And then for part four, we'll actually invite a researcher that has utilized Fresh in their research and talk about their applications and what are their experiences with Fresh bioprinting. And so you can find us on Twitter and LinkedIn, and encourage you to sign up. Before we get started, a little bit about myself. My name is Andrew Lee. I am the head of bioprinting and co-founder at FluidForm. I did my undergraduate studies at UC San Diego bioengineering, went on to pursue a PhD at Carnegie Mellon University in Professor Adam Feinberg's lab. My thesis was focused on really exploring the use of bioprinting for engineering some of the complex architectures found in muscle tissues, with this goal of leveraging these biomimetic architectures that we fabricate to really recreate the core functions of the organs that we want to create. And then so as part of this work, I was really very fortunate to be on a extremely collaborative team of researchers. And together we were able to publish a lot of our work in 2019 in the journal Science. So in this webinar, I will share with you a lot of the images and data from this work, since this is really the core technology that FluidForm as a company is founded upon. And together, I think it conveys our vision of propelling bioprinting towards becoming key to regenerative medicine, tissue engineering, and biomedical research as a whole. So today I want to talk to you about really looking at bioprinting and tissue engineering as a field through a very different lens, right? So when we think about bioprinting, there really is a lot of promises and allure to that term that's been put out there, and there hasn't been too much evidence of us being able to achieve what we say it can be achieved. So hopefully in the next 30 to 40 minutes, I can share with you a different way to think about bioprinting research, why we think that is important, and why without this viewpoint, maybe we won't be able to grow as a field as rapidly as we want, and why this shift in mindset is really so critical that will take us towards impactful research that then becomes clinically relevant. So at FluidForm, our goal is to really be the first to print organs. I know that is an extremely ambitious goal, but we think that we have the technology to do so. And there's a couple of challenges associated with getting to this goal. I think really these two key problems are, first, if many of you on this call are probably working with a unique material that has certain desired properties that you want, and that leads to a certain function. This could be, for example, an electroactive material that can be integrated with excitable tissue. It could be a tunable stiffness material that allows you to control the microenvironment of the cells that you want to study. But most of these materials turns out actually to be very difficult to print or assemble in 3 dimensions. So as a result, a second problem actually emerges, and that is that there is it's not possible to actually fabricate many of the desired geometries using these materials that you might want to use. And so what we've done at FluidForm is actually now made it possible to print these material in any geometry. And this is exactly why I spent all this time in my early PhD years, really, I spent a lot of time trying to print hard to print materials with very limited success. And, I think I really experienced firsthand how a single technology could change the way I thought about my bioprinting project as well as changed my project's trajectory as I really start to develop and integrate this technology into my own work. So I promise I will talk about what we've done to make all of this possible. I like to show this print here to highlight this capability first. This is a scaled down 3D bioprinted model of the human heart that we printed entirely out of collagen 1. This is a derived from a MRI file and then printed using collagen 1 as our material of choice. Some of you might be asking, what exactly is 3D bioprinting? Where did it come from? And how come I'm really just starting to see it at every single conference. There's sessions devoted entirely to 3D bioprinting and biofabrication. So shown here are some pretty recognizable images of 3D printed constructs. On the left, you have a vase printed out of thermoplastic. And on the right, there's a variety of printed metal components. All these parts are really produced using 3D printing processes that have been developed around the intrinsic properties of the materials. So on the left, in a hot glue gun-like process, thermoplastic can be melted and pushed out of a nozzle onto a platform, and then very rapidly solidified upon cooling to build an object layer by layer. And on the right here, we have metal powders inside a container that can be melted all together by a strong laser and sintered together to create a very complex 3D printed parts. And so for a really long time now, a lot of people in the field of biomedical engineering have asked the question, could we borrow these same 3D printing technologies that allow us to print plastics and metals and print biomaterials that are interesting for the human body? And I think the answer to that question has been, a yes and a no. Yes, we can actually borrow some of these hardware and software that have been developed over decades, but no, the biomaterials that we might want to print are just not amenable to being printed. So, these biomaterials that I talk about are oftentimes very soft, right? And we're talking about a lot of these very familiar biomaterials that are used in tissue engineering, such as cells, collagens, fibrinogen, matrigel, decellularized extracellular matrix. All of these listed on the left are very soft biomaterials. A benefit that plastic and metal printers take for granted is that the materials they print are a lot of times far harder than they are soft, than the soft water-laden hydrogels that make up our body, right? Plastics and metals can be heated and cooled quickly to form them into stable shapes. However, the protein hydrogels that make up your body, the same really cannot be said, right? So an analogy that we like to make a lot of times is this watch assembling problem. So if I were to give you this disassembled switch watch, you can probably rebuild this. And this is because while each of these individual parts are small, they're all very rigid parts. And with some time, some knowledge, and some practice, you can most likely learn to put everything back together despite the very complex arrangement of the parts. But if you imagine these parts are now soft, they become very hard to assemble. And if they're intertangled with each other as well, the level of complexity drastically increases. And you might end up with the clocks seen here in this famous Salvador Dali painting where everything slops and droops all over the place. And this is exactly what we see when we go to print a collagen 1 solution onto a dish directly in air onto a platform, right? So here we're supposed to be printing just a 10 by 10 by a couple millimeters of rectangular slab, right, with sharp corners and then also defined edges. But you don't see any of that. Everything kind of coalesces and pulls together all together. And then so with each of these materials on the left-hand side here, they really come with their unique printing challenges that are related to their rheological properties, their gelation mechanism and gelation kinetics. And you really need to wrangle all of these things together in order to get your print to look somewhat decently like a print. So this is really the first core problem that makes bioprinting so difficult. And what we've done as a field is to look at each of these materials individually, assess what makes them difficult to print, and modify the materials for printability to make them printable. So, for example, you can imagine, this print that we previously showed. It's a very difficult to print soft biomaterial. What a lot of people have done is to modify the material chemistry itself to tune the material gelation and mechanism and kinetics, right? So for example, incorporating UV cross-linkable material so that upon extrusion onto a platform, you're rapidly cross-linking by UV light. Another approach is to modify the material composition to then tune its rheological properties. So adding in rheological modifiers so that it's capable of standing up in air and capable of being stacked layer by layer on top of each other. Another way of addressing this problem of printing a very soft biomaterial is that you can print additional scaffolding material to provide your soft material with structural support, right? So each of these things really come with their unique challenges. So for example, when you're modifying material chemistry, you lead to oftentimes reduced cellular binding domains, and then also increased hydrogel stiffness, and then also a lot of times reduced biocompatibility with the modification to the materials. When you're adding in additional components to your ink or to your material, you're lowering the concentrations of all components in the mixed material as a whole. And then when you're printing these scaffolding materials, you really need to then think about how do I deal with the presence of these very stiff structural components that I don't really want in my construct, but they are necessary as supports, right? And then so, because we need to so finely tune each material so that they become printable, we usually don't have quite the perfect printable material. We usually add an in-between point where materials are somewhat printable, but they need to be printed in simpler geometries that are designed around the challenges of printing them, right? So, this is our second problem. You have these desired geometries that cannot be printed with your materials because these materials are so soft. And what we end up with are shapes and geometries that, while they're recognizable, they're not really the ones that we intended to print to start with, right? They're not the complex tissue, lifelike tissues structures that we really wanted to print that got us excited to begin this work. So to summarize these two core problems, you have these unique materials that are hard to print. And secondly, you have desired geometries that cannot be printed with your materials. And you might even notice that we haven't even started to consider the functional properties of the constructs that we're printing, which is really to us the core of why we're doing what we're doing, right? Creating constructs that are functional. And so, usually as engineers, our design process starts with outlining the functional criteria for the thing that we're trying to create, and then picking the materials and geometry. But I think currently in bioprinting, our process is actually kind of reversed. We're spending a lot of time making a material printable, and then we're spending time finding a geometry that is printable with our material, and then we're assessing the function of what we're creating using this specific material and geometry combination that really limits our space, our imagination, and what we can actually create. And so frankly, we want to change that thought process. Right, so before we dive into how we're doing that, I want to kind of invite you on this brief journey, really a thought experiment that might be kind of fun, right? If you were to imagine all these problems that we discussed didn't exist, and you can print anything that you might possibly want to print, what would you print? So, what would you print if you can print using the right materials in the desired geometries, and then really explore the printed function of the construct or the tissue that you create, right? So some pictures here, it's just spark some imagination. You can potentially create small, capillary scale vasculature found here that really helps perfuse the entire organs. You can then scale this up towards the arterioles, and then also then entire full organ scale vasculature systems. The bone is another example of very different scales of architectures and patterns that are so critical to function that if you're able to create the trabecular structure inside the core of the bones and then also the cortical bone, you can start to study some very interesting things like the biomechanics of the bone. How do we load, and how do we create bone-like constructs that can be loaded in different directions? What about a composite of all of these different tissue types, right? So really starting to study how multiple tissue types such as muscles, tendons, ligaments, bones really organize and function in relation to each other. It's also possible to think about printing devices that replace native tissue function. So, such as a heart valve shown here, right? The heart valve itself could be a 3D bioprinted piece. And it also has a core function that it needs to interact with the surrounding tissue in order to become a functional piece that's implanted into a human body. And then you might also really have, start to design and dream up new geometries that doesn't even exist in the body. It kind of just exists inside a computer software that you can then turn into a print that has specific functions that maybe excels beyond what the human body can do. So these are really the scientific questions that we're interested in studying, and the ones that we hope that the world might develop answers for as a result of being able to create these constructs. So, we think we have developed a technology that now allows us to start print at this level of complexity and to really actually ask these scientific questions. So, the technology platform that allows us to print with soft materials in any geometry for function is one that we call FRESH, right? So it stands for Freeform Reversible Embedding as Suspended Hydrogels, or FRESH for short. It's really an entirely new kind of 3D printing known as embedded 3D printing. In embedded 3D printing, we're no longer printing in air, but inside of a support material that cushions our soft ink while it gels in place. So this is the exact same layer by layer printing method that occurs just like it does in plastic FDM printing, only this time it all happens inside our support material. So, this on the left here is an image showing kind of the overview of the process. You have an extrusion syringe containing our soft material bioink. This is being pushed out of an extrusion needle into to create this object here. We're printing the logo FluidForm. And this all occurs inside of our support material, which is the core to this technology. The support material has this unique behavior called it's a yield stress material. So this is similar to a lot of materials that you encounter in everyday life, such as toothpaste, peanut butter, ketchup, mayo. All of these are all yield stress materials, and that means that you can actually squeeze these things out of a tube because when you apply a force, they'll flow like a liquid. And when you leave them alone, they'll actually stay in place because there is this yield stress that needs to be overcome. And this is the reason why your toothpaste doesn't just fall off your bristles when you turn it upside down. And then so our support material consists of these gelatin microparticles that really lends it this yield stress behavior. When all of these microparticles are packed in and jammed tightly together, it lends it this property. So all of this is actually pretty simple and intuitive when you watch it happen. In this video that I'll show here, we have a syringe loaded with a blue ink, alginate, which a lot of you might be familiar with. And then also we're printing into a support material containing calcium ions. So calcium ions help facilitate alginate gelation. And so as we're depositing our alginate into our support material shown here in the container in yellow, we're simultaneously cross-linking and gelling the alginate in place to create our three-dimensional construct. And by the time we're done printing, we can actually melt away our support material because it's made of gelatin. It will melt at 37 degrees Celsius. And we can then retrieve our completed fully cross-linked print. And so this is what allows us to really think about printing with any gelling material, print in any geometry that we want to, and start to print for function. That again, I think that is core to all of this. And so, we initially described FRESH in 2015, published in Science Advances. We then followed this up with a publication in Science in 2019. And this is where, FluidForm has exclusively licensed this technology to really think about how we can leverage this technology ourselves for creating the tissues and constructs for research, repair, and replacement for our internal purposes, but also think about how can we help facilitate the best use of this technology to the rest of the research community. So for these next slides, I want to show you some images and data from our work to highlight what is possible to print it using FRESH. So first, I want to talk about this idea of orthogonal gelation mechanism. So orthogonal meaning that they have distinct gelation mechanisms. And so here I've highlighted 4 different materials: collagen, methacrylated HA, fibrinogen, and alginate. Each of these have very different gelation mechanisms. Collagen will gel when its acidic condition is neutralized and the temperature is raised. Methacrylated HA is gelled by exposure to UV light. Fibrinogen is an enzymatic type of gelation where when you add in thrombin, you can cleave the fibrinogen to turn it into a fibrin gel. And then allogen, as we talked about previously, will gel in the presence of ionic conditions, mainly calcium ions. And so this all happens inside a support bath. And this is where it gets very interesting. Within the support bath, we have these gelatin microparticles, and surrounding these gelatin microparticles, there's this interchangeable fluid phase that is critical for allowing us to print with multiple materials all at the same time. Because in this fluid phase, we can actually start to think about incorporating a pH neutral buffer. We can shine a UV light towards the support bath. We can add in thrombin. We can also add in calcium ions. And so you can start to see why all of these could occur within the same support bath. And then so shown here on the right, we actually printed 4 materials on a single printer in a single print. So this all happened with the 4 materials that we described, and it all happened inside a single print. And then next, we kind of, wanted to think about how do we build the basic building blocks that are necessary for building larger scale and also very finely high resolution features. So we took collagen type 1 as an example material to really explore this, right? Because collagen is the most abundant protein in the human body and really has such a large footprint in biomedical research to understand its role in biology and disease and leverage that understanding for therapeutics. So here we decided to print collagen type 1 filaments. So we went from 200 micron filaments all the way down to 20 micron filaments. And we're able to pack these laterally, stack them, and then very easily change the spacing between these printed filaments and start to assemble these filaments in 3-dimensional structures to produce these macro scale structures. And then leveraging these building blocks, we then start to think about how does this hold up in a larger print over multiple length scales, from the high resolution details to the organ scale structures that we might be interested in down in the future. So we took an STL model derived from a human MRI file, turned it directly into a print. So here shown here is a printed heart model, roughly the size of a large strawberry, printed entirely out of collagen type 1. And you can see that it really recreates a lot of the fine details of the file itself. When we take a cross section of the print, we start to see that we recreate some of the trabecular structure and also some of the infilling structure. I think you can better appreciate that here we took that heart and then actually image it in micro CT. And we show that we can see the right and left ventricle. We can see the trabecular structure, as I talked about before, some of the rectangular infill structures, even the valves, the pulmonary and aortic valve, complete with their leaflets. And heart was really just a scaffold. It didn't have any particular function other than as a demonstration of the printing capabilities of FRESH using collagen type 1. And then so we then set out to really think about recreating function in some of the key components of the heart, with the first one being the valve. Right, so we took a valve STL file and then we went ahead and printed it out of collagen 1 again. Shown here are different views of a printed heart valve in solution. And so here we show that the valve, when taken out, it can actually be handled. The leaflets are patent, it's intact, and then you can actually see that, as a finger will go through and try to manipulate some of these leaflets, that the leaflets are manipulable. And we can actually put this under pulsatile flow to then really start to study how does our valve hold up under physiological conditions. And this is actually one of the key components that our company is currently pursuing, is to start to grow and optimize this valve towards something that is deployable and plantable inside human body. And the next critical piece to the puzzle of engineering heart tissue is that the heart, like any other tissue and organ in the body, requires a dense network of vasculature that supplies blood to its cells. So one of the key challenges in tissue engineering has been the ability to fabricate this vasculature network in 3 dimensions with control over organization and geometry of these vessels. So again, we took MRI data. With MRI data, there's this caveat that's, it actually can't pick up a lot of the high resolution details, such as the capillaries that you might find within your heart structure. So we actually took the MRI data and computationally generated some of the smaller vessel networks that aren't resolvable in MRI. And so we then segmented it out and took a piece of this lattice, this vasculature network, and we actually just printed it. So you can see here, our print resembles very much so like the original design file. And here we actually took an optical imaging approach where we optically cleared our print and we perfused a blood mimic through our vessels, our open lumens, through the larger vessels all the way down to the couple 100-micron scale vessels. So you can see that all the vessels are perfusable and they remain patent and open after printing. Of course, a very key functional component to the heart is this contractile activity, right? That allows the heart to pump blood, which is orchestrated by the highly organized heart cells, cardiomyocytes. So here we created a simple model of the left ventricle, which is responsible for pumping oxygenated blood throughout your body. So we printed this left ventricle model out of collagen and stem cell derived cardiomyocytes. And you can see here through a cross section of the wall, we have distinct layers of our collagen printed surrounding this central shell or central layer of cardiomyocytes. You can see in green and blue, magenta showing collagen. And when we take a closer look at these cells, we find that these cells not only have been printed, they also remain highly viable, actually aligned to the direction of printing, and also elongated and well connected in a dense network. When we then look to study the function of our printed ventricle using calcium imaging to observe the electrical activity of these cells, we find that the electrical activity propagation of the calcium wave cap propagation really propagates throughout the entire surface of the ventricle that we printed, and it's a homogeneous propagation across the surface. We can also look at this from top down to see that anisotropic propagation. And then we can also point simulate this ventricle at a certain point to then start to study under control conditions its conduction velocity and isotropic ratio, all of which we've quantified. So here is just an isochrome map of this point simulation process. And so this left ventricle model is actually really exciting because this is a first step towards creating a three-dimensional in vitro model that could potentially be one day beneficial in investigating how can we develop more effective drugs for that aren't cardiotoxic, but also how can we develop more effective drugs that are that can be treated towards certain diseases. So we can create healthy ventricle models as well as diseased ventricle models. Again, this is something that we internally are working on as well. So we've really seen FRESH grow as a biofabrication platform over the last 5 to 6 years. We've seen a number of very interesting publications from, University of Minnesota, at UCLA, and even our own work showing a large, full-scale human printed heart. This is exciting to us because our philosophy is really trying to make this technology as accessible to the research community so that people can start dreaming of things that we ourselves just don't have the capacity or the time to actually create. And I think the research community as a whole is definitely much more imaginative than us as a small company. So at FluidForm, we also been thinking about, how can we take our technology towards eventual long-term goals of organ scale transplantations? And we've, created some of these parts as demonstration pieces, but I think it kind of illustrates where our vision is. So we've talked about the heart valve. We talked about, we can also create something like the ACL, which is one of the most common orthopedic surgeries out there to replace ACLs. We can see that not only can we handle these printed pieces, we can also flex and create torsion in these constructs. We can also print a pediatric scale trachea, showing that the lumens remain open as features that are on the surface of these prints, and then also it can be bent and twisted and the lumens remain intact. And then also, we also print an intervertebral disc, one of the reasons why a lot of people have back pain, right? When these discs rupture, the central region of the fluids is this fluid jelly-like material starts to leak out and cause inflammation in the body. And so we can start to mimic some of the softer regions of the print of the actual intervertebral disc by creating regions with lower filling density, and then some of the stiffer regions around the outside, the annulus fibrosus around the outside. And so, FluidForm as a company has taken on this mission to really expand the reach of FRESH, because we truly believe it can have significant and lasting impact in biomedical research. So, our support material is commercially known as Life Support, which is really simply a sterile powder that you rehydrate into support material that you previously saw, and you can go from the sterile powder to then printing an object within 15 minutes. What I really want to emphasize here is that FRESH is really simple to adopt and slot into existing bioprinting research workflows. So as I mentioned throughout this talk, FRESH now makes most biomaterials printable, biomaterials that we discussed previously. You can really now explore any geometry limited only by imagination. It's compatible with any printer out there that you might have access to. Next week, we'll talk about how you can accomplish FRESH using one of our partner printers, Lulzbot, that we developed together with them. And then also and then it's also possible to utilize any software that exists out there on the market. So you can see that the workflow is very easy to slot into your existing workflow. And so, I want to include my email address here if you have any further questions about anything bioprinting or fresh printing related, and I'd be happy to chat. So at this point, I just kind of want to open up the webinar to any questions and be happy to discuss further. Let me go to the questions. Give me 1 second. Okay, I have our list of questions here. One of the questions that I see here is, let's see. Is this process compatible with decellularized ECM? And I think the answer to that question is, this process is compatible with a lot of different materials. With decel ECM, a lot of these ECMs will gel in stable conditions, meaning that it's pH neutral, and also it's very similar to collagen in that sense. It needs to be pH neutralized, and then also with response to temperature, so body temperature. So this is actually very easy to accomplish. We print it with a wide range of ECM materials. And so that is something that we've done pretty comfortably before. And this is, again, the process of why we think this platform is so powerful, is that you can start to really play around with your different design parameters and also with your ink combinations. As long as your ink will gel, you can put your ink, mix different inks together, as long as they have this gelling condition that can occur inside the support bath. You can print with stuff like polyacrylamide, PEG, alginate, fibrinogen that we talked about previously, as well as cells. I see another question here asking, what type of printer would you recommend printing with? And what is the best, out of the different optical, extrusion-based, and inkjet printing, what would you suggest? I think that is, like I said before, FRESH is compatible with a lot of different hardware systems. It's most compatible with extrusion-based printers, and one of the reasons, or any printer that has a syringe that can be sucked down into the support bath. And with extrusion-based printers, we're very familiar with them because they allow us to have the most control and highest produce prints with the highest feature resolutions. And then also, it's also doable with pneumatic systems, because pneumatic systems will have a your cartridge hooked up to a needle that you can then put into the support bath. Again, the support bath is really the magic of where it all happens. As long as you can print into the support bath has the property of holding your prints in place while it gels. So I think it's, to answer that question, it kind of is flexible, and you're allowed to really leverage a lot of different existing printers out there. Let's see. Yeah, I see a question here asking about what are some of the applications to the ventricle model. As I talked about before, the ventricle model is really interesting. One of the interesting data that I didn't share is that the ventricle that we printed actually showed arrhythmogenic behavior. So this is something that got us really excited, because this behavior can't typically be observed in a lot of the other preparations that exist out there. These meaning, 2D preparation, 2D cell culture model layers, organoids, and also even tissue strip like 3D engineered hard tissue constructs. And so these arrhythmia is something that we hope in the future we can actually design and control for, and we can start to reproduce these arrhythmias to then study their behavior, and also to understand how they manifest structurally and compositionally inside our in vitro model, and how do we develop a drug regimen for treating these arrhythmias. So that's that the broader application here is that we have the opportunity to create these in vitro models for studying drug safety and efficacy, which I think a lot of you might understand is a critical problem right now, because it's so expensive to develop any drug through the entire pipeline, because it involves so many steps of in vitro testing, animal testing, to human clinical trials, all of which a lot of times they don't actually translate results well from one to another. So I think that's just another that we've that we're really excited about and are currently working on. I think a last question here asks about when do you think organ printing will happen, and do you find that realistic? Well, for us, we've kind of set this ambitious goal for ourselves, and we actually think it's doable. I think it's probably on the far side of 10 years, and that is that's something that we're excited to work towards as we start to develop the technologies that we need to move towards that goal of organ printing. Right now, we can't do it, but I think it really, the challenge here is that we need to integrate, or I think the opportunity here is we can integrate a lot of existing technologies that exist out there, right? So things like machine learning, AI, existing CAD programs that we can modify, designed specifically for printing the structures that we want to print. And also start to leverage some of the advances in biomaterials development, as well as integrate the biological innovations in stem cells and stem cell technology, differentiation technology. And I think what is going to be very unique is that we have the ability to start to integrate all of these technologies into our platform to then hopefully lead us towards a path that translates towards organ printing, and that's something that we're very excited to be working on currently and have the opportunity to be working on. And then, so I think with that, I just want to thank everyone for coming, and just want to remind everyone that every Thursday for the next 3 weeks at this same time, we will be having the next part of our webinar series. And then also make sure that you go follow us on LinkedIn and Twitter so that you have the chance to sign up for the next parts. Thank you so much. Please follow up if you have any questions, and looking forward to seeing you next time.